So far in genetics, we've always been looking at genetic crosses involving the inheritance of only one trait. So that is a monohybrid cross. So it might be a black guinea pig mating with a white guinea pig and all the offspring are black. And there the only trait we're looking at is fur color. It might be a plant with pink flowers having a baby with a plant with white flowers and all the offspring are pink. For example, again, there the only trait we're looking at is flower color. But in reality, we know that offspring get more than just one gene from their parents. Otherwise, we'd just be like a black hole of space for all the other genes and there wouldn't be anything for the phenotype. So you didn't just get the genes or the alleles for blood type from your parents. That means all you'd be is blood. You got the genes and alleles for skin color, hair color, eye color, metabolism rate, etc. So when we're doing a dihybrid cross, it's just one step further than a monohybrid cross. It's still much simpler than it really is, because you have the inheritance of thousands of traits in real life. But here we're looking at a cross where you're dealing with the inheritance of two independent traits. So that's why we call it a dihybrid cross. And the reason I'm saying independent or separate traits is that they do not influence each other in any way. So if we look at examples of these kinds of traits that we'll look at, it'll be looking at the inheritance of seed shape as well as seed color. These two are completely unrelated, independent traits. That means that you can have either of the seed shapes, round or wrinkled, pairing or being present in the offspring in the phenotype with either of the seed color, green or yellow. So it doesn't necessarily mean that the dominant traits always go together or the recessive traits always go together. You can have the dominant traits for color, but then the recessive trait for shape. So that's what we mean independent or separate traits. So we could also look at flower color and plant height, completely unrelated. Coat color for an animal like a guinea pig and coat texture. We always use these contrasting characteristics. So these are either or. So the shape will be either round or wrinkled. Color will be yellow or green. Flower color will be, let's say, purple or white. Height will be tall or short, etc. So there's not a range of phenotypes. So this is what we call discontinuous variation, where there are only 1, 2, or 3 options for phenotype. Continuous variation results in a range, emphasis on the word range, of phenotypes. So like height in humans, there's thousands of different heights if you round it down to the nearest hundredth of a millimeter, for example. It's not like everyone is 1.6, 1.7 or 1.8 meters tall and that's it. The range of phenotypes is continuous variation. The contrasting characteristics of either or traits is discontinuous variation. So a final reminder, homozygous, remember the other word we use for it is pure breeding. So that is a clue given to you in the question. Heterozygous means it's a hybrid. So the reason we tell you those terms, either homozygous, pure breeding, or heterozygous or hybrid, is to help you determine the genotype, to know if it's two dominant alleles or a dominant and recessive allele. Some key things to remember for a dihybrid cross. For each trait that we're looking at, whether it's plant height, seed shape, seed color, the individual always still has 2 alleles for each trait. So that would either be homozygous dominant, so with a big R for shape of the seed, let's say, that's round, or heterozygous, big R, small r, or homozygous recessive. But it will also have 2 alleles for seed color, either homozygous dominant, so it would be a yellow seed, heterozygous, still a yellow seed, or homozygous recessive, which would be a green seed. Because if we just remind ourselves, these 2 alleles are found on homologous chromosomes. So this would be a homologous pair of chromosomes with the allele for, let's say, seed shape. So this would have an allele for round seeds and an allele for wrinkled seeds. But phenotypically, they would have round seeds because capital R, round, is dominant. On a separate pair of homologous chromosomes, there will be the gene and the 2 alleles for seed color. So it's not like individuals are only inheriting the gene for shape of seeds and then they just have no color in the seeds. They will inherit the gene for every single trait about that plant. But they always have 2 copies of the alleles because of the pairs of homologous chromosomes. There's one allele on one member of the chromosome and the other allele will be present on the other member of the homologous pair. So what you end up having is 4 letters making up the genotype. So what I've written here is the genotype. There will be 2 alleles or letters for seed shape and 2 alleles or letters for seed color. So the 2 R's are for the shape, the 2 Y's are for the color. And you can have any combination. You can have both being heterozygous depending on what is present on the homologous chromosomes. One trait can be heterozygous for seed shape. The other trait can be homozygous, recessive in this case. It does not matter the pairings because these are completely independent traits. So now, when we're looking at gamete formation, this is a little bit more complex. You can end up having, when these homologous chromosomes split, this parent can either give a big R to the gamete or a small r to the gamete, but it can also give a small y and a small y to the gamete. So just like with a monohybrid cross, the gametes only have one allele, so one letter. So if we looked at this again, it could either have a big R or a small r, or a small y and a small y, or if it had a big Y and a small y, it would have either. But because we're dealing with 2 traits here, you end up having 1 letter for each trait in the gamete. So the individual can get a big R being in a gamete with a small y, for example, or a small r with a small y. It depends on how gametes form. So there's still 1 letter per trait. There's 1 R and 1 Y. But the only complication comes in because we're looking at the fact that there's the gene for shape of the seed and the gene for color. But there's still only 1 allele per gene. So if we look at how we do that, let's pick this as the most complex example because it's heterozygous for both traits. So you're going to have the most combinations of gametes. If you only allowed 1 R and 1 Y, that R could end up in a gamete with the capital Y. So you could have an allele for round seeds and an allele for yellow seeds in the same gamete. Or this allele for round seeds could pair with the allele for green seeds. So the big R could be with a small y. Same thing can happen. You can have a small r allele for wrinkled seeds going with the allele for yellow seeds. You could have a small r with a big Y or a small r with a small y. So that's how you do gamete formation. You've got to look at what are the combination options for the R and the Y. What are the different options that you can get in the gametes to make sure you have 1 R and 1 Y each time? Doesn't matter if you put the R or the Y in front, as long as you keep it regular the whole time. And here, I've kept R in front. I don't put a capital Y in front, even though that would make grammatical sense. I keep the R in front so that I'm keeping the same format. And the last important thing with regards to dihybrid crosses is Mendel's third and final law. We've dealt with the law of dominance, where in the heterozygous condition, the dominant trait shows and the recessive one is masked. We've dealt with the law of segregation, which is true here again, where the different factors or alleles that control the gene separate or segregate during meiosis, so that each gamete gets 1 copy of an allele. So there's 2 R's in the parent and only 1 R in the gamete. That's true for dihybrid crosses as well. Same with the law of dominance. But Mendel's law of independent assortment is only true for dihybrid crosses because we're looking at 2 traits. So what this states, and it's a difficult one to state, but we look at the fact that the various factors, now we call them alleles, controlling the different traits are separate entities. They do not influence each other. So the alleles for shape of the seed do not influence the alleles for color of the seed. So that means they sort themselves out independently during gamete formation. So you can have a big R going with a small y, or a small r going with a small y. If we look at this example here, there's so many combinations of gametes. Big R with a big Y, big R with a small y, small r with a big Y, and small r with a small y. It is completely random how these gametes form. It's another method that leads to genetic variation in meiosis. We have random arrangement of chromosomes at the equator. So whether these line up this way around or the other way around, this is independent assortment. Don't get those two confused. They are very, very similar. So what we're saying here is round seeds can either be green or yellow. You don't have to have the dominant traits going together all the time. Same with wrinkled seeds. They could also be green or yellow. So dihybrid crosses are the same principle as monohybrid crosses. It just comes in here with the formation of the gametes that gets confusing, that you have to have one R and one Y, but you've got to look at the combination options that are possible. For something that's simple like this, all of the gametes can only have a small R and a small Y. So you just write that out. It'll be like that gamete 4 times. If you watch my next video, I'll show you one example doing a Punnett square using a dihybrid cross. That's also a little bit complex.